Hi there. Can you hear me okay? Can you let me know in the chat, please? Perfect. Thanks, Lana. Okay, so welcome to session 2 of the digestive system and the liver. I'm just going to pop my camera because bandwidth, but let me get the slideshow. Okay, so last session we finished off some questions. And I said we'd start off on some questions this session. So this might help you remember what we were doing last session. So have a look at these questions, pop the answers up in a minute. So have a think and see if you can remember it. What do the villi and microvilli do in the small intestine? Which are responsible, or which substance is responsible for the emulsification of lipids? And what is the function of peristalsis, the process of peristalsis? So have a answers up in a moment. You can write in the chat if you want to. Worry if not, I'm going to put the answers up regardless. Okay, good work. Right, I'll pop the answers up then for those of you at home. So, what do the villi and small intestine? Well, these are structures on the small intestine. They increase the surface area for the absorption of food. The emulsification of lipids is bile. So salivary amylase is an enzyme that breaks, pepsin is an enzyme that breaks down proteins in the stomach. It's yet, but it helps control our blood glucose levels. Also, but we'll talk about that a bit more later on in the course. And what is the function of peristalsis? Well, that pushes the food all the way along the digestive tract. So we had a look at regards to the esophagus, that's this tube that goes from your mouth or oral down to your stomach. But it's also the process that pushes food along your all the way around, along your large intestine, and even is important for egestion, the waste, the feces at the end of the process as well. So it's a really important muscular muscles that go around the tube and the longitudinal muscles that go down, makes up our digestive tract. Okay, so next, but I'm just going to quickly recap of last session because I feel like we're a little partway through the journey from our mouth from last session. So what we talked about last session is ingestion. We put the food into up. The chewing process will increase the surface area of the food. It will cause us to release saliva as well from our salivary glands, and it increases and lubricates it, mixing it with the saliva. So it is physically, mechanical, but also the chemical process of digestion begins because the saliva can chemically break down the food, making it into smaller subunits. Digestion of starch begins in the mouth, and we break the starch down to but 2 units of glucose, and we call those 2 units, it's a dies sugars, we call that maltose. So we swallow the food, called a bolus, and it moves down into that long tube. You can see there on the right of the screen. And the process of peristalsis pushes the food along this tube. So peristalsis is, as I was just saying a moment ago, is the alternate contraction of the tube and the relaxation of the longitudinal muscles below that sort of squeeze point. The food, the bolus of food, lubricated now down to the stomach. There's a little sphincter muscle at the top of the stomach, which regurgitating the food once it's in the stomach. The stomach is a very muscular sac to hold the volume, but also it will mix it with hydrochloric acid, which helps protect us from bacteria and it's also full of enzymes, protease enzymes, which break down. So at this point, we've broken down some starch partially and some protein. And from there, from the stomach, the food moves into the small intestine there, which is a really long tube and has a huge for absorption of food. So the food gets absorbed in the small. It's really, really long, as I said, but it's highly folded. Lumen of the tube is folded as well. And that folding increases the surface area. And on the surface, there are villi and microvilli. And these further increase the surface area and allows us food. We also saw at the end of last session that the diffusion distance is really short between the. And so this bit here is the whole of the tube of the small intestine. It's only 1 cell thick between the lumen of the tube and the blood supply. There's a really good blood supply. This makes sure there's always a concentration gradient. See why that's important today. So short diffusion distance, so nutrients to move, 1 cell thick, good concentration. So there's always a difference in concentration between inside the lumen and in. There's always going to be more sugars or whatever in the lumen and less in the blood. And we've got this large surface area because of all of these tiny little villi and structures that help increase the surface area. And we're going to talk more about those. So from the small intestine, this bit here, the really good blood supply that we were just looking at, which then lead to the liver. And go to the liver via this vein here, and it's called the hepatic portal. So the first place that anything we absorb goes to is the liver. Our liver actually gets really damaged from alcohol consumption or excessive alcohol consumption. Place where that blood goes to. So those capillaries here inside the villi, which is the structures that we were looking at, they drain into the hepatic portal vein, which goes to the liver. So some substances are broken down in the liver, some toxins such as alcohol. Substances are converted into other things. For example, excess protein. This stuff, as long as it's not too poisonous and is useful, is in the blood. The blood is now containing nutrients and it's taken off to where these nutrients become assimilated. So they become part of our cell. Proteins can get, or amino acids can get rebuilt into proteins, built into structures within the cells. So far we've ingested our food, we've absorbed our food in the small intestine, and following on and processing in the liver, these components become assimilated with our cells. And what is left in the small intestine the useful stuff has been taken out, moves then into the large intestine, which is in green. It's not green in real life. What happens in the large intestine is absorbed so that our feces isn't watery all the time. We also any sodium or vitamin K, and holds on to the undigested material. That's basically fiber. So, whoops. Eaten gets mixed in with bacteria, but we can't digest it. There are a lot of bacteria that live and intestine. We think that the appendix is probably a repository here. It's labeled up over here. Of it because appendicitis is something that happens fairly. It's a very painful condition, and people die from a burst appendix. It is pretty serious, but we think that appendix, we use vestigial organ, like it's got no use anymore. It's just something that's there from our. There's a few things like that. For example, the embryo has gills during its development from our. Obviously, we don't have gills anymore, but you can still see it in our embryonics. Used to think that the appendix was a vestigial organ also, but actually it's probably a repository for bacteria and helps us to add some sort of illness. It helps us to repopulate our guts with awful lot of bacteria live in our gut. They're really important. They help us break down. We are beginning to realize they have a role to regulation of our immune system as well. We don't understand it fully, but protect us a lot from our own immune response and help to regulate. So what's happening in terms of the food waste? We've out of it pretty much now, apart from a bit of water that gets reabsorbed, of our mostly the fiber and bacteria that we're getting rid of, compacted and moves down to the rectum, which is down here, and then is expelled via the anus by contractions. So the same peristalsis that helps us swallow our food, esophagus to our stomach, up here helps us to expel the waste at the bottom. Important about this expulsion of waste, we call it egestion. It's not excretion waste from your body. We only excrete stuff that has been metabolism. So with the waste that is removed from the digestive from our metabolism, we shovel that in through our mouth and it comes out the other end. It in our body is just the stuff that we couldn't use, that we ate earlier on. But when we breathe out and excrete carbon dioxide in our body through biochemical processes, and when we excrete in our urine, we've made that through processes in our body as well. So we're excreting stuff that we've made, but we ingest stuff that we've one end and comes out the other end. So let's have a little look at and see what we can remember or recall. Which part of the small intestine is the stomach? Can you remember which part comes first? Okay. So we've got the jejunum, the ileum, and the jejunum there. They'll neutralize in the small intestine. And what happens during the process of... So have a little look at those. You can pop the answers up if you want in a moment with you. But do have a go, even if you're not too sure. Okay, well done. I'll pop the and you can compare and see. So the first part of the small intestine is called, and what does bile neutralize? It neutralizes the acidic stomach contents and assimilation. Nutrients are taken around the body and used to make new structures. What is the colon? Oh, yeah, sorry. The colon is the large intestine. It's another word for. So we have the ascending, transverse, and descent. Basically, that just tells you which part. Look, so there we go. Means going up. Transverse, which means going across, and going down. It's just the large. You can describe regions of the large intestine. Sigmoid colon, S shape, and the rectum and anus at the bottom there. So yeah, they're just parts of it. Very rarely used unless you were at the doctor's and they were describing part of a illness or something like colitis, you know. So very, very rarely used, to be honest. So last bit part of the system is the rectum, which is part of the large intestine. It's the last bit stored before egestion. And the anus is another sphincter muscle. Remember, top of the stomach, which stops us vomiting constantly. It's there as a to stop you losing your feces constantly, because that would be not very pleasant. So come over. Which process is digestion? What is the main site of action for enzymes that we've been looking at in the last session? And where is the feces stores before it is egested from the system? Just have a look over and see which ones you can recall. I'll pop those answers up for you. Oops. Right. So egestion does not involve, but to excrete something, it has to be a metabolite. It has to have been made in your body, like carbon dioxide. Yeah. We're removing the undigested material, the fiber. Defecation, which is the process where peristalsis, alternate contraction, the feces out. Main site of enzymes is the small intestine as a whole. Probably it's a little bit more than other areas, I think, but small intestine anyway. Digestive system stores the feces before egestion. It is the rectum. That's the last little bit of that tube. Okay, so quick summary of the process. We have ingestion, which is where we put the food in the mouth. We have digestion, which is breaking down the food into smaller soluble subunits. Soluble so that it can get moved across the small intestine. So food is digested initially in the mouth and then the stomach and then finally the small intestine. The process can be mechanical, such as chewing or the churning in the stomach, or chemical using enzymes. We then absorb the small soluble subunits of the food from the blood into the small from the small intestine. And then these nutrients are then assimilated. They're taken around the body and used up in the cells, where they become part of the cell structure. And the last part of the process is egestion, where we remove the fiber, really, which we cannot extract anything from. There's nothing left that we can remove from our food at that point. So, quick look through some questions. Which is the correct order of the processes? Where does digestion of carbohydrates begin? Where does digestion of proteins begin? And where does the emulsification of lipids take place? Have a quick think through and see what you can remember. Okay, I'll just pop the answers up for you to have a look. So the process goes ingestion, digestion, absorption, assimilation, and egestion. And digestion of carbohydrates begins in the mouth with the enzyme salivary amylase, which breaks down starch. Digestion of proteins begins in the stomach with the enzyme pepsin. And emulsification of lipids takes place in the small intestine with bile, which is produced in the liver, stored in the gallbladder, and released down the bile duct into the small intestine. Remember, that's increasing the surface area of the lipids so that the lipase enzymes can work on them. Okay, I'll skip past that so we can get to the next bit. So what have we done so far? Well, we haven't had a look at osmosis yet, but we have done point number 2, looking at ingestion, digestion, absorption, assimilation, and egestion. We have had a look at the structure of the villi. We still need to look at diffusion and osmosis. We're going to do that in a minute. We've had a look at some examples of the digestive secretions like bile and their functions in the different regions of the human gut. We've also had a look at some enzymes from salivary amylase to pepsin and some of the secretions of the pancreas, trypsin, chymotrypsin as well. So any questions before we move on and start to have a look at movement across the cell membrane? I'll just get the next bit or slide up while we do that. Are chyme and the bolus the same? No, not quite. When we talk about the bolus, we're talking about the food which has been chewed up in your mouth and swallowed. When we're talking about chyme, it has been through the stomach or is in the stomach, so it has picked up the secretions of the stomach as well. So it has got that gastric juice and the hydrochloric acid and stuff as well. It's just the saliva and obviously the secretions that are in the saliva and the food when it's a bolus of food. Okie doke. Let's have a look then next. Pop the next lot of slides up for you. Right, so what we're going to have a look at now are processes called diffusion, osmosis, and active transport. These are ways of moving stuff across the cell membrane. It's quite important in the digestive system because we were just talking about absorption. And to do that, you need to move stuff across the cell membrane from the lumen of the digestive tract into the blood. So we're going to carry on with digestive system, and we're going to start with looking at diffusion. So what is diffusion? Well, it's the movement of particles from a region where they are in high concentration to a region where they are in low concentration. So that sounds quite technical, but in reality, it's quite simple. Here we've got some particles. They're in a region of high concentration, and they're going to spread out to a region of low concentration. That's all it is. So over here, there was a low concentration of red particles. They will spread from their high concentration until they are in equilibrium, fairly equally spread out. We're all very familiar with this. Think back to primary school when you put your paintbrush into the nice clean water, you would see diffusion happening. That's all it is, nothing more complicated. It will happen without any energy. If you heat it up, it happens a lot quicker because all of the molecules start to move around because of kinetic energy. But it will happen anyway, just because of physics. Okay, so the particles spread out until there's an equal concentration throughout the container. It's a passive process. No energy is required to make this happen. They will just spread out. Really, what's happening is they're all moving in random directions. They don't all whoosh over to this side of the beaker at all. They're going up, they're going across, they're going down, they're going into each other. But eventually they collide with each other and the water particles so much, and that just spreads them out into that space. Diffusion is very important in living systems. It's how we get oxygen into our lungs. The oxygen is in a high concentration in the air compared to in our blood. So it moves from where it's in high concentration in the air to where it's in low concentration in our blood. And the converse happens for carbon dioxide. That moves from where it's in high concentration in our blood to where it's in low concentration in the air. So the lungs are an exchange surface that rely on diffusion. We've just been talking for the last session about the digestive system, which is also an exchange surface. So both the lungs and the digestive system are adapted to maximize diffusion. Happens in leaves as well. It's how leaves get their carbon dioxide and oxygen in and out for photosynthesis. So what we can use diffusion for is moving molecules across the cell's membrane. So this is the cell membrane that we had a look in the first session. Remember, we said it's a bilayer or a double layer of molecules, and there are proteins embedded in it, and some of these proteins can cross the entire membrane, and they can actually act as channels to move things across. And one of the things that we move across is water. So when we move water across a membrane by diffusion, it's got a special name. We call it osmosis. So osmosis is just a type of diffusion. Again, we're moving something, but it's water this time, from a region where it's in high concentration to a region where it's in low concentration. But it's also got the caveat that it's got to be across a membrane that's partially permeable, essentially a cell membrane. So osmosis is the movement of water from a region where it's in high concentration to a region where it's in low concentration across a partially permeable membrane. So again, that sounds a bit technical, but all it's saying is water molecules will spread out as well, and they will move across membranes. So let's have a look at what this means in terms of a cell. So this is the extracellular fluid at the top. So that means extracellular means outside the cell. And this is the intracellular fluid at the bottom. So intracellular means inside the cell. Everything's watery. It's just how it is in biology. Nothing really survives well without water. So inside the cell is watery, outside the cell is watery. And we've got a partially permeable membrane here. What do we mean by partially permeable? Well, all it means is some stuff can get across, some stuff can't. Think of a sieve or a colander that you use when you're cooking. Those are partially permeable. water can get through, pasta, rice, whatever can't. Yeah. So it's small enough for some very small things to get through. Small soluble things we were talking about quite a lot last session. We wanted to get our food, proteins, carbohydrates, starch broken down into small soluble molecules. Small soluble stuff can get across the membrane a lot easier. So partially permeable, so it's permeable to small soluble stuff like, I don't know, maybe amino acids or glucose, but not big molecules like maybe proteins or starch. So in this diagram, I've got some water molecules, which look a little bit like teddy bear faces with ears on, but that's just the way a water molecule looks. That's the way the atoms arrange themselves in water. You'll learn about that, I expect, when you do the chemistry modules. So water, does anyone know the formula for water, the chemical formula? Don't overthink it. Let's just have a quick look. H2O, yeah, exactly. So H2O. So what colour is my H then, do you think, on my water molecule that I've drawn up there? It's green, H2. So 2 H's and 1 O. Yeah. So that's all it means. I've got 2 H's and 1 O. What do you think that H and that O stands for? If you had to have a guess. Hydrogen, good. And what's the O then? Oxygen, good. That's all it is. Very simple molecule, hydrogen and oxygen. So we've got our H, our 2 and our O, and H2, H times 2, 2 hydrogens and our O. And what you can see here is we've got a concentration gradient on the outside of our cell membrane. So we've got a lot of water. So we've got a high concentration of water outside the membrane. What's going to happen is it is going to move across the membrane. It will move back and forth in both directions. It's not constant, you know, it just takes a while. And it will keep moving until it's equally distributed on both sides of the membrane. Now, let me just go back to that before I start running that animation. So we've got a equilibrium there that we've reached. So that's osmosis. Are we meant to be at 80% water? Yeah, probably somewhere around that. It's pretty high. I think it might be 79 point something, but yeah, it's pretty high. Extracellular fluid and extracellular matrix. Yes, matrix implies that there are some fibers and stuff there. So probably but yeah, extracellular fluid, it just means it's outside the cell. Yeah. Right, let's have a look at this. This shows osmosis. So again, we've got a cell membrane, double layer, inside of the cell, cytoplasm, and outside of the cell, we've got solutes. So the solute is some sort of salt or sugars. There's all sorts of stuff dissolved in our cells. So let's go back. There you go. What happens? We've got the solute outside the cell. High solute concentration outside the cell. You can see there's more salts and a low solute concentration inside the cell. So the water is going to go from where it's in high concentration, which is inside the cell, to where it's in low concentration outside the cell. You can see on that cell membrane, there's some protein channels that the water can move through. They're called aquaporins. So the water is going to move from where it's in high concentration originally to where it's in low concentration. So let's look again. High concentration of salts outside the cell. So that means that there's a low concentration of water. I'll explain why in a minute. So there's a high concentration of water inside the cell compared to outside. And that's going to mean the water is going to move from where it's high in concentration inside the cell to where it's low in concentration outside the cell. And it will move through these aquaporin proteins. No energy is needed for this process. It will just happen because of the laws of physics. There's a concentration gradient initially. So high salt outside the cell, so that means there's low water. I'll explain why in a minute. So low salt inside the cell, so that means there's relatively high water. So that high water concentration inside the cell means there's concentration gradient. The water is going to move from where it's high in concentration inside the cell to where it's low in concentration outside the cell. So that's just osmosis. Water's moving from its high concentration, in this scenario, inside the cell, to a low concentration, in our scenario, outside the cell. So let me just explain why we have because it's a lot of salt outside the cell. You can see here, this green molecule is salt or solute. So as a result, our water concentration is low outside the cell. And that's because all of our water molecules, these are these red and white molecules on this diagram, are associating with a salt molecule. So they're not free water molecules. So even though there is water out there, there's not free water molecules. They're all associated with the salt. So even though in a moment you'll be able to see the water molecules drawn outside the cell, there is a lower water concentration because all of those water molecules, none of them are free. They're all going to be associated with salt, giving a lower concentration outside the cell. Essentially, all the water molecules are tied up because of that high salt concentration. So that's giving us a concentration gradient where there's loads of free water inside the cell because there's not much salt. There's hardly any free water outside the cell because there's loads of salt. And it's going to cause the water to move from its high concentration inside the cell to outside. So before I talk to you a little bit about concentration, do you have any questions about that osmosis? Because I know it's a little bit tricky. Some people find it a bit challenging. I'll carry on talking while you're thinking about it. What we have here is a beaker of water. It's distilled water. So that means we have a high concentration of water. There's no impurities in it. It's just water. We say that it's really dilute. What I've got over here is loads of salt. It could be sugar. It doesn't have to be sodium chloride salt. It's just salts. So we've got a low concentration of water. You can see there's still the same volume of water there, but the water molecules are associated with those salt or sugar molecules. So there's not so much free water, even though it looks volume-wise like there's loads of water. If you looked at a molecular level, the water molecules would be associated with the salt molecules. So we've got a concentrated solution over there. Now, this can have certain effects on cells. Here you can see some blood cells. And what happens if we put blood cells in a solution? Don't worry too much about these terms, isotonic, hypertonic. But if we put blood cells in a solution where there's equal amounts of salts and water inside the cell and outside the cell, they just look normal. If we put them into a solution that's got loads of water in and not much salts, the water will move into the cells because there's less water in the cells compared to outside, because all the water inside the cell is tied up to whatever sugars and salts that are inside the cell. So the water will move from a high concentration outside the cell to a low concentration inside the cell. And the reverse happens if you put blood cells in a salty solution. All the water moves from inside the cell where it's in high concentration to outside the cell where it's salty, because the water's in low concentration there. So isotonic solutions are when the solution and the cell are the same, and then the distilled water will cause water to move into the cells from high to low concentration because it's in lower concentration in the cell. That can actually pop the cells. It lyses the cells. So that's obviously quite dangerous in animal cells. Doesn't happen in plant cells. They've got a cell wall which protects them. Animal cells have no cell wall to withstand the pressure of the water moving in, and they pop. So we skip that. Any questions about osmosis and diffusion so far? Okay, so this is quite a key topic, and I feel like it can be a little bit complicated when you first go through it. So it's one to get your head around, make sure you understand, because as you move on with this course and obviously whatever courses you're choosing to do next year, this is an important topic to have your head around. So make sure you get it. Okay, so as we were talking about earlier, one of the things that we saw with the villi and microvilli that really helps to move stuff by diffusion is surface area. So things that help increase the rate of diffusion include a large surface area, the larger the better, a short diffusion distance, which we saw in the fact that there's only a 1 cell thick barrier between the lumen, the inside of the digestive system tube, and the blood. We need a good concentration gradient because that's what drives the process. The higher the concentration, the better. We can also really increase diffusion with temperature if we're looking at it from a purely chemical basis. So if you were in a chemistry lab and you needed to increase the rate of diffusion, first thing you do is heat it. Not such a good idea in biology, obviously, because you'll start to cook tissue and kill cells, so it doesn't work. But the reason diffusion is important to us is because we're large multicellular organisms, so we have a huge volume. We can't get oxygen in like smaller microorganisms can just absorb all of the oxygen that they need through diffusion. We can't. We're massive multicellular organisms, so we need exchange surfaces like the lungs and the intestines, and fish use gills, for example. And the reason for this is that volume increases at a much faster rate than surface area. If you think about the formula for volume, like this cube, it's got length of each side. Well, it's just written as algebra at the minute, like S. So it's width times height times depth, or S times S times S, or S cubed. So that's volume, but the area, as you can see here, the net of the cube, the surface area, there's 6 faces to a cube. Think of a dice. There's 6 faces, and you need to find the area of each face, each square face, and multiply it by 6. But so let's imagine if this square face of the cube had 2 centimeter length, so it'd be 2 centimeters times 2 centimeters would give you the area of 1 square, so that would be 4 centimeters. There's 6 of them making up the whole cube, so 6 times 4 is 24. So that surface area of that cube would be 24 centimeters squared. But if we then did the volume, it'd be 2 times 2, which is 4, times 2 again, which is 8, and that's 8 centimeters cubed. So because it's cubed, the volume increases much faster than the surface area. We'll have a look, compare that now. So volume and surface area are not increasing at the same rate. Large organisms have to adapt these specialist areas, the lungs, the gills in fish, the intestine, to get in the nutrients that they need. So let's compare some cubes then. So surface area of this 1 centimeter cube, there's 6 faces and they've all got a 1 centimeter squared area. So it's got a surface area of 6 centimeters squared. Its volume is 1 times 1 times 1, so 1 centimeter cubed. So it's got a surface area to volume ratio of 6 to 1. Now, this cube has got 2 by 2 centimeters length. So its surface area is 24 centimeters. So if we do 2 times 2, that gives us the area of a face. 4, 6 faces, like die. So it's 24 centimeters squared. But its volume, 2 times 2 times 2, is 8 centimeters cubed. So surface area to volume ratio, 3 to 1. And then if we look at a much bigger cube, which has got 3 times 3 centimeters length. So if we do 3 times 3, that's going to give us 9 centimeters squared per face. 6 faces, so that's 54 centimeters squared. And if we work out its volume, 3 times 3 times 3, 27 centimeters cubed. And that now has a surface area to volume ratio of 2 to 1. So as you can see, we go from a surface area to volume ratio of 6 to 1, to 3 to 1, to 2 to 1. So the bigger our cube is getting, the smaller the surface area to volume ratio is getting. So if these cubes were cube-shaped animals, the bigger our animal would be getting, the smaller its surface would be in relation to its volume. Yes, the surface area is increasing. Obviously, it's getting bigger, but it's not increasing as fast as the volume. And that's because the surface area is squared and the volume is cubed. So the volume is increasing at a much faster rate. So we go from surface area to volume ratio of 6 to 1 in the small organism, 3 to 1 in the medium organism, and 2 to 1 in the large organism. So imagine my organism is getting its oxygen and glucose in by diffusion. I mean, this one's got an okay surface area to volume ratio, might work out okay and survive. This one, probably dead. This one, definitely dead. Surface area is way too small to cope for that big volume. We can have a look at this from a side-on view. Let's imagine we are going to submerge my cubes into some liquid and see how long it takes for this liquid to diffuse into the middle of these hypothetical square cube organisms. So we're going to leave each of these organisms to soak, and imagine this is a glucose solution, and the whole organism has got to get glucose to all of its cells to survive. Come back an hour later, the pink dye containing our glucose nutrients has diffused the exact same distance. So this organism, it's not too bad. It's covered a reasonable proportion of the volume. This one is just the outside. This one is hardly touched it. So that's why we need to have these big exchange surfaces to speed up diffusion, like the small intestines and the lungs. Just check. Isn't that why lots of different animals have specialist features to help them alive in different climates? That's another type of adaptation. It is an adaptation, but it's specific adaptation of organisms for their habitats. These are more general adaptations of organisms to help them cope as multicellular organisms. So think of these as sort of tier 1 adaptations that you have to have. You can't specialize unless you've got a functioning gut and lungs, and then we can worry about, have we got proper insulation for the snow or big enough feet so that we don't sink in the sand and things like that. So this is like first level adaptation. This just helps us be, yeah, exactly, stage 1. This just helps us be multicellular rather than some sort of primitive worm, basically. This helps us have access to food and nutrients, and then we can worry about camouflage and stuff like that. But yeah, you're right, they are adaptations. Surface area of volume, right? So surface area is literally the area on the surface of the cube. Whoops, what happened there? Hang on, sorry about that. Okay. So the surface area is this that you can see here, the net of the cube. It's just the flat surface area. So not the space inside the cube, just as if you were covering the cube in wrapping paper or something. How much wrapping paper would I need to cover it, or wallpaper? It's just the surface of the cube. So it's flat, it's an area. Yeah. So if I said to you, find the area of a square that had 2 centimeter length, you'd find the area is 2 times 2. If I said find the area of a square that had a 3 centimeter length, you'd just say, well, that's 3 times 3. So that's all it is. Because we're looking at a cube, it has 6 faces, like dice that you roll to play board games. So we have to find the area of 1 square and then multiply it by 6. That's just the surface of the cube. And then if we were finding the volume, that's the depth as well. So it's not just the area on the outside, it's the depth of the cube too. So if I said find the area of a cube, you would find the area of 1 face and then multiply it by 6. And if I said find the volume, you would multiply the length 3 times. So look at these. Okay, the surface of this cube, we've got 6 faces, and each face is 1 times 1 centimeters. So we do 1 times 1, and that's just 1, and there's 6 faces. So it has a surface area of 6. This cube has 2 centimeters by 2 centimeters. So that's 4 centimeters squared face, and there's 6 faces. So we do 4, because it's 2 times 2, 4, multiplied by 6. So that's got an area of 24 centimeters squared. And then this one here is 3 times 3 centimeters, 9, 6 faces. So 9 times 6 is 54. So does that make sense, how I've got these surface areas on these cubes? Can you see what I've done? If not, that's fine. Just let me know. Why is the volume a smaller number? Well, it's, if you just follow the maths, it's 1 times 1 times 1, which is 1. And then if you do this one, it's 2 times 2, which is 4, times 2, which is 8. And then if you do this one, it's 3 times 3 times 3, which is 27. But if you keep it increasing the length of the side, you'll see that the volume gets bigger quicker. So if you carried on doing the maths, you'd see that the volume would get bigger quicker. The reason I've used these numbers is so that my surface area to volume ratio is easy to follow the maths. So what's happening is the volume, because it's, if you look, cubed, it's a cubed value, it's going to get bigger quicker. And the surface area, or the area covering the cube, is squared, so it doesn't get as big as quick. So the volume, although the surface area increases, obviously, because our cube is getting bigger, so it is going to have bigger surface area, but the volume increases much quicker, at a much quicker rate. and that's because it's a cubed value, and that is just a squared value. But this time we're multiplying more bigger numbers together, if you like, because one of the numbers that we're always going to be multiplying together to get the surface area of a cube is going to be 6, because it's always got 6 faces. So that keeps it quite small. I think probably the most important thing, rather than the maths itself, because you can always go over the maths, is this slide here. So if you imagine you've got each one of those cubes and you put it inside a beaker, and I've just got some pink dye so I can track how far it's diffused. And it's when you look at this, it's really important, because the distance that the dye diffuses is always going to be the same. So on a small cube, proportionally, it gets much nearer to the center of this small cube than it gets to the center of this much larger cube. So that's where the importance is of this concept. It's really hard to diffuse something into the middle of a big organism. That's why we need lungs and digestive system, for example, to help with that exchange of substances. So yeah, it is smaller because of the area. Sorry, I'm just reading the chat now because it said, why is it a smaller number? Because of the area. It's the relationship between the surface area and the volume that's important. So it is how big is the surface area compared to the volume. So it's not just the absolute number, it's the ratio, it's the relationship between the surface area and the volume that's important. So that's why we're constantly talking about surface area being really important, increasing the surface area with the gut having the villi and the microvilli. It increases the surface area. We're always trying to increase the surface area for diffusion. At the lungs, when we move on to talking about them, we'll be talking about increasing the surface area there as well. So in that case, more dye would be needed. No, more dye won't make any difference because it's just the surface area that we've got to work with. We can put as much dye in as we like. It won't make the slightest bit of difference. What we can do is we can increase the time that we're soaking it for. That would help. But if that was a real life scenario and these were cells in the middle of this cube that were relying on oxygen, they might well be dead by the time that diffuses in. I mean, this smaller one probably survive because it's got a smaller volume to cover. This one would just die. The cells wouldn't survive in the middle because adding more liquid is not going to increase the surface of area that is available for it to diffuse through. So it wouldn't make any difference. So yeah, but if you imagine if we deconstructed this, and rather than having it as a 3 by 3 by 3 cube, but we flattened them all out, so we had just one big flat rectangle, that would massively increase the surface area. And if we separated out all the individual 1 centimeter cubes that were making up this big cube, that would even greater increase the surface area. So that would improve our diffusion into these cells, if you like. So it's all about the surface area that's in contact with this pink dye. That's what's important. And with these bigger volumes, it's just not effective. I'll just skip through that. Now, the last thing that we can consider to move stuff across the cell membranes is active transport, but we're a little bit out of time to do that today. So we'll have a look at that next session. But this is a really important trick in biology because we can use energy. to move stuff across the cell membrane. So we'll have a look at this next session. So have a think if you've got any more questions on surface area, and I can give you a bit of support with that next time. Okay, thanks everyone, and I will see you next Tuesday. I think the slides are up for you. I'm pretty sure I put them up, but yeah. Bye, guys.